It's for me a great pleasure to introduce all of you Noemí Esteras. Noemí is graduated in pharmacy by the Universidad Complutense de Madrid. And she did her PhD at the Centro de Investigaciones Biológicas at the CSIC, under the supervision of Ángeles Martín Requero. She then carried out most of her research career as a postdoc and later as a senior research fellow at the Queen Square Institute for Neurology at the University College London at the UK, in the group of Andrei Abramov. In 2022, she obtained a Ramón and Cajal fellowship and joined in 2023 the lab of Javier Fernández Ruiz at the Universidad Complutense in Madrid. She has recently obtained funding from the Ministry of Science to develop her own research line as an independent PI. Her main line of work is focused in understanding the interaction of mitochondria, oxidative stress, and calcium signaling in neurodegeneration, and in particular in the pathogenesis of tau-related frontotemporal dementia. She's also interested in the role of NRF2 as a modulator of mitochondrial function, both in brain physiology as well as in therapeutic strategies in neurodegeneration. Her talk today is titled "The Different Roles of Mitochondria as Modulators of Calcium Signaling in Tauopathies." So, Noemí, thank you very much for being here and for your willingness to share with us your work. And the floor is yours. Please go ahead. Thank you.
Thank you. Thank you so much for your introduction. I'm going to share my screen here. I hope you can see it correctly.
Yes.
Yeah, okay. Thank you so much. So, well, thanks a lot for the introduction and for the opportunity to present part of my work here. So this work that I'm going to present today is mostly what I did during my postdoctoral stage in University College London, which was focused on trying to understand the role of mitochondria in the pathogenesis of tau and the interaction, as you said, between the different functions that mitochondria have, and especially that related to calcium signaling. So, as all of you know, the microtubule-associated protein tau is a soluble and intracellular protein that in the central nervous system is mostly present in neurons and particularly in axons. The main role that we all know about tau is that it's involved in the microtubule stabilization. There are 6 different isoforms of tau that are generated during neuronal development, and broadly we can speak about 2 different types, the 3R and the 4R isoforms, depending on how many microtubule domain regions they have. The 3R isoforms have 3 binding domains, while the 4R isoforms have 4 binding domains to the microtubules. Tau is involved in different neurodegenerative conditions called tauopathies. In these disorders, we find that the position of misfolded tau in brain inclusions, together or not with other proteins. These disorders are clinically very heterogeneous. Probably the one that we all know is Alzheimer's disease, but there are other dementias and also other movement disorders that also present the position of tau in the specific areas of the brain. They are characterized by a distinct neuropathology, both related to the area where they are found and also to the isoform of tau present in the isoforms. For example, there are tauopathies where only 4R tau is deposited. There are other tauopathies where only 3R is deposited. There are other tauopathies where both are found in the inclusions. The involvement of tau in the development of disease became clear when mutations in MAPT gene, which is the gene encoding tau, were found to be causative of frontotemporal dementia. There are different types of mutations leading to different biological effects. The one that we studied during my postdoctoral stage in University College was the 10+16 mutation. This is an intronic mutation that causes an increased splicing in of exon 10. In the adult normal brain, we have a similar amount of 3R and 4R isoforms. They are in a balance. However, with this type of mutation, we have an increased splicing in of exon 10, which encodes one of these microtubule binding domains. So when we have this mutation, instead of having a balance between 3R and 4R, we have an overproduction of 4R tau. And this imbalance, the increased overproduction of 4R, is enough to cause neurodegeneration. So we wanted to understand the pathogenesis of this mutation, how tau is involved in the mechanism of neurodegeneration, in particular in these events. So we worked in very close collaboration with the Hospital of Neurology and Neuroscience, and we had access to samples from patients from the neurogenetic clinics. And in collaboration with Selina from University College and Professor John Hardy, we had access to fibroblasts from patients harboring these mutations. By reprogramming with retroviral transfection of the Yamanaka factors, we generated iPS lines, which are induced pluripotent stem cells that we can then differentiate into any cell type of the body that we are interested in. In our case, because we were interested in studying the pathogenesis of dementia, we differentiated these cells into cortical neurons. The protocol we followed, this was done a few years ago. Now there are faster and more efficient protocols than this. It was based on a SMAD inhibition. And after more or less 90, 120 days of differentiation, we were able to get mature cortical neurons. We characterized them by electrophysiology. They were all very well characterized. So we had access to samples from 2 different patients with this 10+16 mutation in MAPT and 2 lines from healthy controls. And in addition to this, we generated an engineered iPS line in which we introduced the 10+16 mutation in tau gene in a parental line. This way, we were able to get an isogenic control to understand if the differences that we see in the patients versus the controls were related just to the mutation or to any other type of problems that the patients may have. So the experiments I will show you have been done in all these cell lines. All the key experiments have been replicated in the different models to make sure that it was actually tau which was causing the changes I'm going to show you. In addition to these endogenous models, we also did a model of extracellular tau, because tau, it's an intracellular protein, but it can be secreted and it can be uptaken by neighboring cells. We did in previous experiments some experiments that show that extracellular tau also has a role in pathology. So in addition to the endogenous models in human neurons, we did primary co-cultures of neurons and astrocytes that we treated with extracellular 4R tau. In particular with this fragment, the K18, which has the 4 microtubule binding domains. Because as I told you, this mutation increases 4R tau, causes an overproduction of 4R. And we are interested in understanding what's the role of this in pathology. So we were basically a mitochondrial lab. We were interested in understanding what's the role of mitochondria in the development of different neurodegenerative conditions. And as you all know, mitochondria are involved in almost every aspect of the cellular life. These are vital organelles. The main function that we all know is metabolism and energetics. We know that mitochondria harbor the electron transport chain and all the complexes necessary for ATP production in oxidative phosphorylation. And this is, of course, vital for neurons. Also, because of that, they have an increased ROS production. Mitochondria is the main source of ROS inside the cell. But also, they have other functions, such as calcium signaling. This is essential as well for neurons, because in neurons which have electrical activity modulated by calcium signaling, mitochondria are able to shape these signals by uptaking calcium and releasing calcium. So they can buffer and modulate the calcium signals from the cytosol, and this is essential for neurons. And also, they have an important role in starting the process of apoptosis in specific conditions. So what we have studied is how all these functions that mitochondria have interconnect between each other and how they have a role in the mechanism of neurodegeneration induced by 4R tau. And these are the results I'm going to show you today. So we first, as with every experiment with mitochondria, the typical experiment we normally do at the beginning is to check the mitochondrial membrane potential. You know that mitochondria, through the different complexes in the electron transport chain, they pump protons into the intermembrane space that they use later to produce ATP through complex 5. So the first experiment we did was to check how was the polarization of the mitochondria. And to our surprise, we found that in these neurons, I will talk indecently about the FTD patients of the engineer line. All of them are related to the 10+16 mutation. So all of them, I will talk indifferently about both. So in all the models that we checked, we had an hyperpolarization of the mitochondria. The mitochondria was more polarized in the patients compared to the controls. And this was somehow surprising at the beginning, because what we normally find in neurodegenerative conditions is the other way around, that depolarization of the mitochondria, so then they are not able to produce enough ATP. So this was somehow surprising at the beginning, that in these cells, and it was replicated in all the models, we had the opposite effect. When we next check the ATP levels, because this is something that we also check to make sure that if they are able to maintain or not the ATP levels, we found that at this stage, the patients that you can see here in red and purple had similar ATP levels compared to the controls. So they had a mitochondrial dysfunction, but ATP levels were maintained in basal conditions. They were maintained in similar levels to the controls. However, when we checked where this ATP was coming from, you know, we have two main sources of ATP inside of the cell. We can produce ATP by glycolysis and by oxidative phosphorylation. In the neurons, the most effective way to produce ATP is oxidative phosphorylation. So they mostly produce ATP this way. However, what we found is that in the controls, while in the controls, they were producing most of the ATP by oxidative phosphorylation, but in contrast, the patients didn't. But they were able to overcompensate glycolysis. They increased the levels of glycolysis, and they were able to compensate this reduction of ATP produced by oxidative phosphorylation. So the result was that the basal ATP levels were similar between both. This was in basal conditions, and this was at a specific age. We don't know what happened later, but at least at this age, they were able to maintain the ATP levels. So one might think that this hyperpolarization of the mitochondria could be something good, right? If the depolarization is something bad because they are not able to produce enough ATP, then hyperpolarization can be a good thing. But actually, it's not, because when mitochondria is hyperpolarized, they normally tend to produce more reactive oxygen species. So the next thing we did was to check the production of ROS by the mitochondria in these cells, and we found that actually they had much higher levels of mitochondrial ROS compared to the controls. We treated them with MitoQ. This is a specific antioxidant for the mitochondria, and we were able to reduce these levels of mitochondrial antioxidants. And importantly, these higher levels of ROS were directly related to the higher levels of neuronal death that we had in these neurons of patients. This was the basal death. It was higher in the patients compared to the controls. But when we treated with MitoQ, with this specific mitochondrial antioxidants, we were able to prevent neuronal death. So this means that in this first part of the research, we found that the metabolism of the mitochondria in this 10+16 mutation was altered. We had an increase in the mitochondrial membrane potential that was leading to higher levels of mitochondrial ROS that in the end caused neuronal death. And importantly, it could be prevented with MitoQ, with a mitochondrial antioxidant. I will show you later how this mitochondrial ROS was key in the disease. But then we moved to check, okay, so how is another of the functions of the mitochondria, the calcium signaling, is it working correctly or not in these cell lines? So as I told you, these are glutamatergic cortical neurons. And in the neurons, so whenever we have electrical activity, at least in this case, glutamatergical activity, we have cytosolic calcium pulses. And whenever we have an increase in calcium in the cytosol, the mitochondria is able to buffer it, they uptake calcium, and then they release it. This is how a control line behaves. We have an increase in calcium in the cytosol, and in parallel, the mitochondria uptake and release calcium, and then go back to basal levels. So we did these experiments, as I said, by applying glutamate, because these are glutamatergic neurons. When we did the same in the neurons of the patients, what we saw is that they had a much higher signal, both in the cytosolic calcium and in the mitochondrial calcium. You can see when we apply glutamate, the calcium signals are much higher in the patients, and they remain elevated for much longer compared to the controls. And it was mimicked in the mitochondrial calcium. They also uptake more calcium, and the levels inside of the mitochondria remain higher for a much longer time. They don't go back to basal levels as the control do. So to understand if tau was somehow affecting the specific role of the mitochondria in uptaking and releasing calcium, we next did some experiments in which we removed the cytosol. These are called permeabilization experiments. So with these experiments, we permeabilize with digitronin the plasma membrane. We remove the cytosol and we replace it by a buffer of known composition by us. So this way we get rid of the interference from the cytosol, and we can just apply calcium and see how the mitochondria, without any cytosolic interference, behave, how they uptake and release this calcium. In the mitochondria, calcium is uptaken by the MCU, the mitochondrial calcium uniporter, and later it's pumped out of the cell by the NCLX. This is a sodium-calcium exchanger. So we pump calcium out of the mitochondria, and at the same time, we incorporate sodium inside of the mitochondria. So when we do these permeabilization assays, when we apply calcium, you can see in black, we have the control. So the mitochondria uptake calcium, and then they release calcium, and it goes back to basal levels. In the cells that were treated with tau, we have a normal uptake. They uptake the similar amount of calcium as the control do, but then they are not able to release it. They are not able to pump it out. And this is suggesting that MCU is working well, but we have a problem with NCLX, with the sodium calcium exchanger. And actually, the tau lines behaved the same way as if we treat with a specific NCLX inhibitor, you can see here. To actually prove this, because we know that they work in parallel with sodium, we are able also to measure in parallel the sodium levels, okay? And when we do this, we have that in the controls, we have like a mirror effect. So calcium goes up and then it goes down, and sodium does the opposite effect because it works in the other direction. However, in the cells treated with tau, we also have an opposite effect. We have that calcium goes in and then it doesn't go out, and we have the opposite effect with sodium. So this means that actually tau, it's blocking the NCLX, the sodium calcium exchanger. And it was described for the first time by our group, the specific role of tau in this exchanger. So what happens if we are not able to pump the calcium out of the mitochondria? Mitochondria will start accumulating calcium. We have a mitochondrial calcium overload. And specifically in a context where we have a lot of mitochondrial rows, as I showed you at the beginning, this can trigger the opening of the MPTP. The MTP is the mitochondrial permeability transition pore. In normal conditions, the inner membrane of the mitochondria is impermeable. However, when we have this pathological condition of high levels of calcium, high levels of ROS, mitochondria open a pore where all the contents of the mitochondria go out. They are released, and the mitochondria swells. And because, for example, cytochrome C is released, we start the apoptosis process. So whenever we have an MPTP opening, we have a neuronal death. And this is often a common mechanism of neuronal death. So to understand, because of course, if NCLX is blocked in our condition, these cells will be more vulnerable to this. They will tend to open the MPTP earlier than the controls because they have higher levels of mitochondrial calcium inside of them. So to monitor this, we employed a specific fluorophore, that is a substrate for caspase-3, so we can monitor in live imaging how fast caspase-3, how fast apoptosis is activated. So we put the cells, both the controls and the patient lines. We did a calcium overload with them with ferrutinin. It's a massive calcium increase in the cytosol, and we can see how fast they trigger apoptosis. In the patient lines, you can see apoptosis. We see this whenever this yellow nuclei appear, is that apoptosis has been activated. So you can see it happens much earlier in the patients compared to the controls. When we do, of course, in both it happens because we have an overload of calcium, but FTD cells, tau mutated cells, they activate caspase-3, they activate apoptosis much faster than the controls. Because of this NCLX inhibition, they are much more vulnerable to any changes in cytosolic calcium. They cannot handle it as well as the control, so they trigger apoptosis much earlier. So yeah, the conclusion of this part is that 10+16 MAPT neurons are more vulnerable to calcium overload. And if you remember, when I showed you the video at the beginning, these cells had not only increased mitochondrial calcium. When we applied glutamate, they also had an increased calcium signaling in the cytosol. So we thought, okay, maybe they have a problem with the glutamatergic signaling. Let's explore that pathway to see if everything is working well, because we have cells that are already vulnerable to any changes in the calcium levels inside of the cells. So we focused on that, on the glutamatergic signaling, and we repeated these experiments. This is also calcium imaging. On the left, we have controls. On the right, we have the FTD lines. When we apply a physiological glutamate stimulus, we see that in the controls, we have an increase, and then it goes back to basal levels. However, in the patients, we have an increase, and then this increase stays elevated for a very long time. And it only goes back to basal when we remove the glutamate from the media. And this was giving us some information. So it's not only because the mitochondria is not pumping and is not handling the calcium correctly, it's also something related to glutamate, because when we remove the glutamate, everything goes back to normal levels. So we wanted to explore also what was happening here in this signaling. So you know that glutamate is the main excitatory neurotransmitter in the brain. And in the neurons, it's able to act through different types of receptors. We have the ionotropic receptors, which are basically channels in the plasma membrane. And we have the metabotropic receptors, which are coupled to G proteins. And they can also be activated and release calcium through the endoplasmic reticulum. So we first wanted to explore, okay, is something happening through any of this pathway, through exactly which pathway it's happening this. So we first to rule out the role of metabotropic receptors, because these ones are able to release calcium from intracellular stores, not from the calcium is not entering through the plasma membrane. It's being released through the plasmic reticulum. We did a first experiment in which we removed the calcium from the extracellular media. We apply glutamate, and we see that when we do this, nothing changes. The patients still have nothing changes. But when we put again the calcium in the extracellular media, we are able to recover the response. So this means that metabotropic receptors don't have a role here. So if something is happening, it's happening through the ionotropic receptors. So we are able to discard that part and focus on the ionotropic receptors. So to see if it was something general, because we have the NMDA, the AMPA, and the kinase receptors, we did first as an approach in which we employed specific inhibitors of these different receptors. And in all of them, we were able to, so we had the specific one for NMDA, specific one for AMPA, and these two that were for AMPA and kinase. And as you can see here in the patient lines, so this is the one without the inhibitors, a sustained, very long, elevated response. Where we inhibit NMDA and AMPA, we are able to recover a normal signal. So this means that something is happening in these lines through the NMDA and AMPA receptors, because when we block them, we are able to recover the calcium signal. And when we do the same, but instead of using the antagonists, we use the specific agonists, we are also able to replicate it. So if instead of glutamate, we use NMDA, we can see that the patient have a higher and sustained response, and the same for AMPA. With kinase, it was not significant. So we know that this mutation, the 10+16 mutation that leads to an overproduction of 4R tau, is able to impair calcium signaling through NMDA and AMPA receptors. I don't have time to show you this here, but we also corroborate this with electrophysiologic experiments, where we could see that the currents were indeed altered through these receptors. So we thought, okay, there is a problem here. Could it be linked to what we have observed before? Because we know, as I showed you in the first slides, that the increased mitochondrial ROS has an essential effect here in triggering the neuronal death, because when we block mitochondrial ROS with MitoQ, we prevent neuronal death. So if somehow we have a missing link here between oxidative stress, cell death, could this be somehow affecting this signaling through glutamate, this calcium signaling? So to do this, because we knew that MitoQ was excellent in preventing the neuronal death, we repeated these calcium experiments and we put the cells, we repeated the same experiments, but in the presence of MitoQ. So these are patients, regular response, as you remember, high intensity response, sustained for longer. However, when we treat with this mitochondrial antioxidant, with MitoQ, we recover the same signal that we see in the controls. It's a lower intensity and it goes back to basal. So yes, there is apparently a link here that links mitochondrial ROS and this impairment in the calcium response. To corroborate this, we treated the cells with different types of antioxidants, with Trolox, that is a general antioxidant, MitoQ and MitoTempo, that are specific for mitochondria, and then PUFAs, which are working on the lipid peroxidation level. And we saw that both mitochondrial antioxidants, MitoQ and MitoTempo, were able to completely recover the calcium signal of the patients to a similar shape of that of the controls. So it seems that yes, mitochondria is modulating calcium signaling through mitochondrial ROS. Again, we corroborated this by doing some electrophysiology experiments where we saw that reducing the mitochondrial ROS were able to reduce the currents through the glutamatergic receptors. So we thought, okay, how can this be happening? It must be something related. Of course, we have high levels of mitochondrial ROS, we have high levels of oxidative stress. So maybe these ROS are oxidizing the glutamate receptors and we are impairing their functionality because they are oxidized. So to try to have a clue on this, in collaboration with Dr. Morana Jagangak from Croatia, we did some redox proteomics experiments in which we analyzed the whole proteome to check which proteins were specifically oxidized in the patients, but then recovered by MitoQ, because if something like this is happening, we are able to revert it with the antioxidant. So it must be something reversible. It's not something that we cannot change. It's not some irreversible oxidation. So these were the total number of oxidized proteins. Of course, as expected, the patient had higher levels of oxidized proteins, and we had here many proteins that we were able to recover with the MitoQ. When we checked specifically which proteins these were, we were a bit disappointed because we were expecting to have the glutamate receptors here. We were thinking that it could go this way, but the glutamate receptors didn't appear to be oxidized or recovered with the antioxidant. So it was not going through that. We had some enzymes for metabolism, some cytoskeleton, something like that, but it was not the glutamatergic receptors. However, when we checked in the literature, we found that many of these proteins that appear had a role in the trafficking of the glutamate receptors, because glutamate receptors are constantly trafficking between the surface of the cells, between the surface of the plasma membrane and the intracellular side. And there were a few proteins here that were having a role in this process. So we next wanted to study how was this trafficking? How were the glutamate receptors? Were they in the surface? Were they in the intracellular part? We had more glutamate receptors, less. So to do this, we did some cross-linking experiments. And we focused first on the AMPA receptors. I will go briefly through this. We have 4 different subunits of the AMPA receptors. So whenever we have subunit 2, the edited form of subunit 2, these channels are not permeable to calcium. And whenever this subunit is not present, they are permeable to calcium. So when we did these cross-linking experiments to specifically assess how much receptor we had in the surface, we first used this antibody. It's a pan-AMPA receptor antibody that detects the different subunits. And indeed, we saw that in the patients, we had more AMPA receptor present in the surface. And when we treat with the antioxidant, we are able to reduce these levels. So actually, these cells had more glutamate receptors in the surface. The total levels were similar, but the trafficking was affected. We had more AMPA in the surface. And then, of course, when we apply glutamate, we have more calcium entry through them because we have more receptors. We specifically checked as well the subtype 1 as opposite to the subtype 2, and we saw that actually this one, that it's permeable to calcium, we had higher presence in the surface that was corrected when we treated with mitotempo. So we have that predominantly these are calcium permeable receptors that are more expressed in the surface, and that mitochondrial ROS is controlling how much glutamate receptors we have in the plasma membrane. We repeated this with the NMDA receptors. Here, briefly, these are also tetramers, and the subunit 1 is present in all the receptors. So this was the first subunit that we checked. And here we didn't find any differences. Here, tau was not affecting the number of NMDA receptors that we had in the surface, and mitochondrial ROS didn't have a role here. So the total number of NMDA receptors were similar between the patients and the controls. However, we then focused on the NR2B subunit because it was described that this subunit is specifically related to excitotoxicity, which means with calcium-related neuronal death. And in fact, we saw that this subunit was regulated by mitochondrial ROS. We have higher levels of this subunit in the surface, and when we treat with a mitochondrial antioxidant, we are able to remove these receptors from the surface and increase the trafficking to the intracellular side. So similar NMDA receptor expression in the surface, but increased expression of this specific subunit involved in excitotoxicity. So you can see how mitochondria, through mitochondrial ROS, is able to modulate how many glutamate receptors we have in the surface and how much calcium we enter through them. And as a final proof, we did some excitotoxicity experiments in which we induce neuronal death by a very high dose of glutamate. Of course, in the patients, we had higher levels of cellular death, of neuronal death, both in basal and after applying this overdose of glutamate. And what is more important is that CNQX is an AMPA, a glutamate receptor inhibitor. Of course, it's able to block the cellular death. And importantly, MitoQ, the mitochondrial antioxidant, is able to reduce neuronal death in a similar way as the AMPA blocker. So this means that by blocking the ROS produced in the mitochondria, we are able to modulate the calcium entry through the glutamatergic receptors. So this way we have like an alternative tool to try to modulate glutamatergic calcium signaling. So in summary, this is a brief summary of the data I have presented now. We have that in these lines, in the 10+16 mutation in tau, which leads to an overproduction of 4R tau. 4R tau is interacting with the mitochondria through different mechanisms. First, it's able to block NCLX. It blocks the mitochondrial calcium efflux, so neurons are more vulnerable to any changes in calcium. But apart from that, it's able to control calcium through a different mechanism by the overproduction of mitochondrial ROS, which are key in the pathogenesis. We have an increased expression, an increased trafficking, so an increased expression in the surface of specific receptors of glutamate. So we have more entry through the glutamate receptors. We have more entry of calcium in the cytosol. Mitochondria are not able to buffer it as they should because NCLX is blocked. So we have calcium overload both in the cytosol and in the mitochondria, leading to neuronal death. And importantly, both mitochondria and cytosolic calcium overload can be prevented with mitochondrial antioxidants, which we suggest could be a new tool to study in the pathogenesis of this disease. So this was my main line of work that I did in London. And as Miguel said in the introduction, from last year, I am here in the group of Javier Fernández-Ruiz in Complutense. And I have recently obtained funding to continue this project. And basically, the key questions that I ask myself and that I would like to explore with this project is, first of all, how specific is this for the different tau isoforms and mutations? Because, of course, this was all done with the 4R overproduction. We don't know if this is a general mechanism or specific for this. How does tau impair mitochondrial calcium? What is the molecular mechanism? Is it common or not? And what pharmacological strategies can we use to prevent this calcium-induced neuronal death? And I would like to take advantage of the talk and ask if you know anyone that would be interested in joining our lab to do a PhD. They are welcome to send their CVs to these emails here. So I work in the group of Javier Fernández Ruiz, but together with Elisa Navarro, we are starting a new line focused on the role of mitochondria and microglia in the development of neurodegenerative conditions. So if you know anyone that could be interested in doing the PhD, in applying, especially now that the calls are open for the FPU and the Comunidad de Madrid predoctoral fellowship, so please let them know that we are looking for candidates. I would like to thank all my group in London. I work with Professor Andrei Abramov, which is an expert in all this, mitochondria, calcium, and oxidative stress. Elena Britti, we did most of the experiments related to the NCLX. Marta, who also worked in this model, and then Olga and Dimitri, who did the electrophysiology experiments. Selina, John Hardy, and Elizabeth, who did the first steps of the IPS work. Sima Kamar, who generated the 4R that we used for the extracellular experiments, and Morana, who did all the redox proteomic assays. And now thank you to Javier Fernández Ruiz Group, which has hosted me here in Complutense. And well, thanks all for listening. And if you have any questions, I'm happy to answer. Thank you for your attention.
All right, thank you very much, Noemí, for a very nice presentation. So now the paper is open for discussion. As I said earlier, if anyone is interested in posting a questions, please do so by raising your hand using the tool at the Teams. And I see we already have a couple of questions from Carlos Matute. So Carlos, please go ahead.
Hi, everyone, hi. And Happy New Year. Noemí, excellent. I like it a lot. Many questions. Yes, a couple of them. So do you have an idea what are the mechanisms retaining more permeable subunits of the AMPA and NMDA class in the membrane? Do you have a guess, an idea? Are you exploring that?
We haven't explored it in deep. Actually, this is something that we would like to do. So according to the redox proteomics data, it could be something related to proteins involved in the trafficking that are oxidized by these mitochondrial ROS. And these are reversible oxidations that we can recover with the antioxidants. But we didn't, it was just a guess to see more or less how it could go, but we didn't explore it in deep. And this is something that I would like to try.
And the second question, there are other cell types which also have tau, and like oligodendrocytes, and they also have AMPA and MDA receptors. Do you think the same mechanism applies?
This is, thank you for the question. It's a very interesting question. Before leaving London, we were starting to do some work in astrocytes because they are also able to express 4R tau. And we saw some differences. We didn't get into deep into the glutamatergic signaling, but this is, of course, something I would love to explore because it's not only about neurons and tau is not only in neurons. So I think other cell types have a role. And I would love to explore that, how both are interconnected and if it's common or not for the different cell types. Yeah. This is something definitely I will explore.
Thank you and congratulations for the work.
Thank you. Thanks a lot.
All right. Thanks, Carlos and Noemí. We also have a question from Carolina. Carolina, please go ahead. You're mute, Carolina.
Hi. Hi.
All right.
Hi. Okay. Yeah, I was saying that very good talk, as always, Noe. I have a question for you. Maybe we talked about this before, but what do you have some hypotheses about why exactly tau caused this mitochondrial dysfunction? Because you saw all the mechanisms of how what happened in the mitochondria, but what exactly makes tau affects mitochondria and why? I think this is what you are going to explore after. Why, if this is important, is 3R tau or not 3R tau, or what is your thoughts here?
Yeah, thanks a lot. Yeah, this is something we will explore with the project now. We had funding now from the Plan Nacional. We are going to see. Well, I did a first approach with a 3R tau, and it didn't seem to have the same mechanism, but it was just a very preliminary thing. I would like to explore it now with iPS lines with different mutations leading to different biological effects, and see if this is something common or specific. The preliminary results seems that it could be something specific for 4R tau, but we still don't know. It's very preliminary. We will explore if it binds to NCLX, if it's able to oxidize some proteins as well around NCLX. We really don't know. We think it would have, we never explored it in deep, but we think it could also have a role with BDAC. BDAC is a channel in the mitochondrial membrane, and we know that it's related and it can be blocked and unblocked by tubulin. And because tau is so related and so closely functioning in so close relation with tubulin, this is something that we will explore. If we have more or less tubulin that could be blocking or unblocking BDAC, this is one of the mechanisms that we will like to explore as well. But we still don't know. This is one of the objectives of the project.
And do you think that is like a more soluble tau, or is like a more cause of the aggregates? Because you also saw that there was like only the short fragment was causing something similar. So
I think this is more. I think it's something more related to soluble tau, because in the IPS we don't find aggregates. So it should be something related. And yeah, it's something that happens, for example, could happen with synuclein, with other proteins that aggregate. You never know if it's the actual aggregates, if it's the monomers, the oligomers, all the different species. So I think in this case could be more related with soluble tau. I don't know which structure, which specific components, but more soluble than insoluble. That's my guess.
Okay, thank you so much.
Thank you.
All right, thank you, Carolina, for the questions, and Noemí for the answer. Any other questions? Okay, I don't see any other raised hand yet. Let me check the chat. Okay, no more questions coming through the chat either. Let's wait for a few seconds if there is any other question coming in. No more questions? Don't be shy.
Everything was clear.
Yeah. Okay, I don't get any message through the chat. I don't see any raised hand. Okay, so if there are no more questions, then I think it's time to close the session here. But not before thanking once again Noemí for her willingness to present her data today, and also congratulate her for a very nice presentation. I wish you all the luck in the new endeavor that you have here in Madrid, back from London. And I wish you all a great afternoon, and I hope to see you in the next upcoming webinars. All right. So thank you very much, Noemí. Thank you very much, everyone, for attending, and have a great day.
Thanks a lot for having me. It was a pleasure. Thank you.
Bye-bye.
Bye.